Okay, good morning, good afternoon, everybody. Thank you for joining us for the second Brain Matters webinar for this series, Genetic Testing and Genetic Counseling for Dementia with RBC Wealth Management Dominion Securities. My name is Dina Boone, and I'm a public educator with the Alzheimer's Society Southwest Partners. I will be the MC and monitor for this webinar. Susan Oster, another public education coordinator with the Society, will be helping with the technical side of things and managing the chat box. At the Alzheimer's Society, our role is not only to provide programs and services to people who are going through the journey of dementia, but also to provide dementia education to advocate for those living with cognitive disabilities. Today's presentation has been generously supported by RBC Wealth Management Dominion Securities. And before we begin, I just have a little housekeeping business to go over relating to the webinar experience. Please note that your cameras are off and microphones are muted for the webinar. This event is being recorded and the recording will be available in the future on our website. At the end of the presentation, we will have approximately 15 minutes for a moderated Q&A session. We will do our best to address as many questions as we can. During the presentation, if you think of any questions for the panelists, please add them to the Q&A box located at the bottom of your screen. You may have to scroll your mouse around just to find that the icons that come up, and then type your questions in that Q&A box. If you would like to submit a question anonymously, please click the Send Anonymously box before clicking the Send button. I would also like to mention that we have enabled the live transcription option for those of you that would like to have that enabled. There's a little square box with a little CC at the bottom of your screen. If you just click on that and enable that feature, you will see the transcription appear at the bottom of your screen. Any questions that have been submitted prior to the webinar have been shared with the panelists. We have also enabled the upvote feature in the Q&A box, which allows people to see all questions being asked and to move the most popular questions to the top of the list. If you see a similar question posted by someone else, you can simply vote on the question by clicking a thumbs up icon underneath the question, as seen here on the screen. Once the icon is clicked, the question will move up in the list. Should you have any technical concerns, please type these in the chat box and Susan will be happy to help you. You may need to move your mouse around to see the Zoom controls and icons as well. You can submit your comment. Since everyone's situation is unique, the information being provided in this presentation is for educational purposes only and not specific advice for your situation. Should you have specific questions about your situation, please contact your family physician for advice. We now have the pleasure of welcoming our panelists. Dr. Elizabeth Finger is an associate professor and clinician scientist in the Department of Clinical Neurological Sciences at Western University. She is a neurologist at the Parkwood Institute and a scientist at Lawson Health Research Institute. Susan Conacher is a genetic counselor in the Medical Genetics Department at London Health Sciences Center. Susan provides genetic counseling for the Adult Neurological Predictive Genetic Testing Program. And now, please welcome Dr. Finger.
Thank you very much, Dina, and thanks everyone for attending this session. And I'd especially like to thank Susan Conacher, who you just heard is a genetics counselor here in London, for accepting the invitation to present on this topic. I think we're all going to learn a lot from Susan. She's going to start us off. I will speak a bit in the middle, and then Susan will bring it home, and we've saved about hopefully at least 15 minutes or so for questions at the end. So Susan, I'll turn it over to you.
we go. Can everybody hear me now? All right, so I thought I would start off this afternoon talking a little bit about genetics and what it is. So an introduction, Genetics 101. We talk about genes and gene mutations, but people don't necessarily understand specifically what that means. And so moving forward later in the talk, we'll be talking about some sort of technical things and to try and help bring everybody's understanding up. I thought I'd give kind of a brief overview. So all of our genetic information is all of our genetic information is packaged on structures called chromosomes. So the complete blueprint or instruction manual for how to make you and how to make you function is packaged in your chromosomes. And every single cell of your body has the full complement of chromosomes. And there are 46 chromosomes in 23 pairs. And they're paired because you've inherited one copy from your mother and one copy from your father. And each chromosome has hundreds of different genes on it. So that means for each gene that you have one copy from your mother and one copy from your father. So if you take a chromosome and unwind it, what it actually is a tightly wound ball of DNA. And DNA is a spiral staircase looking double-stranded molecule. And our genetic code is encompassed in our DNA. So a particular gene, and remember there are lots of them spaced along this chromosome, will have a start, read through the code, and then we'll have a finish. So I like to think of a chromosome being like a bookshelf full of books. And if you take a book off the bookshelf, the book is comparable to a gene, and you open it up and there are meaningful sentences or instructions that it's providing. So if we open and unravel DNA, double-stranded DNA strand, it reveals the genetic code, and our genetic code has 4 letters to it, CAGT. Our alphabet has 26, but our genetic code just has these 4. And any gene will have a starting code of 3 letters and a finishing code of 3 letters. And then you read through the code, get the building blocks of a protein that's being made by that particular gene. So every 3 letters in the code of a gene codes for the building block that goes into it. So this is the code to say, okay, now you put in the blue building block. This is the code. Now you put in the red building block. Walking along, the next one that's going to go in is the green building block. So what's a genetic change? What's a genetic mutation? Well, any change in our genetic code has the potential to cause variation in the population. So there's lots of variation in our genetic code. Most of it's benign. It's based on our ancestry. It's based on other factors. But in some cases, when we change the code of a gene, kind of like a genetic spelling mistake, it actually changes the function of the protein, and that's potentially a disease-causing mutation. So in this case, you can see that a C has been substituted in, or G has been substituted in. And what that does is it changes the building block that goes in at that position. Or if you insert a letter, now you've shifted all of the code along. And so every building block that gets put in it from there on in is different from what it should have been. And if one of those gets converted to a stop, it means that it will prematurely stop building the protein, and so it will never complete the protein. And that may also be something that can cause disease. Or if you delete a letter, then again, that changes, it shifts the code, and the building blocks that now go into the protein at that position have changed. So what I did is I put it, because not everybody necessarily understands nucleotides and amino acids, I put it into our own alphabet. So if we take the sentence, the cat ate the rat, and we change a letter to make it the cat ate the bat, well, it's still a usable sentence. It still makes sense. Cats could conceivably eat bats. We could change it, the car ate the rat. Well, it's still a readable sentence, but it doesn't make as much sense. If we insert a letter, then what we do is we've shifted the code, and now the letters after where we've inserted the letter don't make any sense. And that potentially could be something that would be a disease-causing mutation. If we insert 3 letters, we may just be inserting a new building block, which doesn't necessarily affect protein function. Again, if we delete a letter, it shifts the code, and everything from there on in is different, and in this case, not readable. And if we put in a letter that causes a stop, the protein doesn't potentially get made. So how do we decide when we're walking along the code of a gene and we see this variation, how do we decide what's relevant? What's disease causing? What's not disease causing? What's just population variance? Well, it used to be these got overcalled, and anybody who had testing before 2015, we tend to reevaluate their variants because it might have been that it was called pathogenic or disease causing when it really wasn't. In 2015, the American College of Medical Genetics came out with some very strict standards in terms of what gets called a pathogenic or likely pathogenic genetic variant. And those are the ones that we're interested when we're doing genetic testing, because those are the ones that have been identified and proven to be disease causing. There's lots of benign variation, so lots of population variation. And then every now and again, we get what's called a variant of uncertain clinical significance. This is the most frustrating finding, because what it means is we don't have enough scientific evidence to know, is it causing disease or is it benign? So it's essentially inconclusive, and it's not useful for assisting with diagnosis, and it's not useful for family screening. So how do we pass our genes on from one generation to the next? Well, let's go back to our chromosome picture. All of our chromosomes in our cells are usually scattered about, and what has to happen at the time egg and sperm cells are being formed is chromosome 1 finds chromosome 1, chromosome 2 finds chromosome 2, and they all line up along the center of the cell. And then there are spindles that pull the pairs of chromosomes apart, and they go to the opposite sides of the cell, and then the cell cleaves down the middle and divides and forms 2 new cells, each with half the amount of genetic information in it. And so what that means is that when egg cells are being formed by the mother, her egg cells divide in half, so each of her egg cells then has 23 unpaired chromosomes, and all of the genes are unpaired now as well. Same thing's happening in the formation of the sperm cells. Sperms have 23 unpaired chromosomes with only single copies of every gene. Egg fertilizes with sperm, you go back to the normal number of 46. And that's how you get genetic information from both your mom and your dad, and it's equal. So half of your genes come from mom and half of your genes come from dad. Mendelian inheritance, there are several inheritance patterns: dominant, recessive, X-linked, and dominant is the key one for us when we're talking about inherited forms of dementia, because to date, the genes that have been discovered that are disease causing, that cause the inherited forms of dementia, are dominantly inherited. And in dominant inheritance, here we have dad that's affected, and D stands for the gene version that has the mutation in it, and little d is the normal version of the genetic code for that gene. So mom has 2 normal versions, so when she's making egg cells, she can only pass on the normal version. When dad makes sperm cells, half of his sperm cells will have the mutation, half of his will be normal. So depending on which egg fertilizes with which sperm, the child is unaffected or the child inherits the mutation. If they haven't inherited it, they don't have it to pass on. If they have inherited it, they will potentially develop that disease, and they are now at a 50% risk to pass it on to any future children they have. Recessive inheritance is a little different. You sometimes hear about people being carriers for genetic diseases. What it means is they carry the abnormal gene or the abnormal version of the gene, which here is represented by small r, but they also have a normal version, which is dominant over that abnormal version. And so the person themselves are not affected by the genetic condition. But if an egg cell and a sperm cell meet that only have the mutation, and that child does not end up with a normal copy of the gene, then they'll have that recessive condition, and that happens about 25% of the time. All right, so that was a very brief overview, but hopefully it gives you enough that when we're talking technology later, it will help you understand. So I'll transfer over to Dr. Finger to talk about diagnostic testing.
Great, thank you so much, Susan. So we'll now move to start to apply some of that general knowledge of genetics to Alzheimer's disease and the other causes of dementia. As we do that, I just wanted to bring us all together in the terminology, and I know for much of the audience this is familiar, but when we use the word dementia, we're using it as an umbrella term that refers to many different diseases that ultimately lead to the loss of cognitive functioning. This could affect memory, judgment, problem solving, and language. And it happens to the extent that it interferes with a person's daily life and activities. When we think about then the genetics of dementia, it's important to consider the specific diseases that can result in that loss of function, because these diseases are each associated with distinct molecular patterns of abnormalities in the brain cells and their own genetic risks. And so we need then to think about them individually when we think about the role of genetic testing. When we see someone in the clinic today and we're considering a diagnosis of dementia, we do want to consider what might be that specific cause. What is the underlying disease? We make that diagnosis based on the following evaluations. Depending on the suspected cause of the dementia, we may or may not talk about genetic testing and whether or not it would be useful. In most cases, genetic testing doesn't change the diagnosis for someone that's been diagnosed with Alzheimer's or FTD or Lewy body disease. Rather, it enables us to see why the patient may have developed that disease. And in some cases, it may lead the patient to consider participating in clinical trials and research trials that are evaluating new mutations that specifically target that gene or that genetic mutation. For a patient with a clinical diagnosis of Alzheimer's disease, most of the time, in fact, over 90% of the time, it's what's considered late onset Alzheimer's disease. So late onset Alzheimer's is the terms used when the onset of symptoms begins at age 65 or older. And thus far, there are really no dominant genetic mutations that are associated with late onset Alzheimer's disease. Only genes that can affect the risk and either increase risk or serve as protective factors, or perhaps multiple genes that all can individually increase or decrease the risk. Because there's no single deterministic gene that can predict for sure whether one gets Alzheimer's or not, at this point, genetic testing is not recommended clinically for people presenting with late onset Alzheimer's disease. Now, the other less than 10% or so of Alzheimer's disease happens earlier in life. And when symptoms come on clearly before the age of 65, we technically use the term early onset Alzheimer's disease. Early onset Alzheimer's really makes up less than 10% of all cases of Alzheimer's disease. And of that 10%, only a small fraction end up having a dominant pattern of inheritance, like Susan referred to. In about 1% of all patients with Alzheimer's disease, we may see a gene mutation in one of the three listed there, the amyloid precursor protein, APP, presenilin 1 or presenilin 2, but that's quite rare. Thus, in patients presenting with early onset Alzheimer's disease, typically we will see their symptoms coming even in their 40s or by their early 50s. It would typically be affecting at least one member of each generation, as Susan showed, so that you get the passing on from parent to child, and each one tends to develop symptoms if they live to middle age. So we only consider genetic testing in patients with early onset Alzheimer's disease if they have other biological family members who have also had early onset Alzheimer's disease. Otherwise, we're unlikely to find any gene mutations that we can interpret. In Lewy body disease, it's quite rare for clusters to occur in families. So usually in a patient with Lewy body disease, they're the only one in the family across multiple generations who's had that. And thus, we do not typically offer genetic testing for patients with Lewy body disease. It has been known for a while, though, that patients with Lewy body disease tend to have a higher than average family history of some who've had Alzheimer's disease or the closely related Parkinson's disease. And just this year, earlier in 2021, there was a very exciting study or interesting study that came out in Nature that found 5 risk genes for what we would consider sporadic Lewy body disease, where we weren't seeing inheritance patterns. 2 of these risk genes were previously known to be risk genes for Parkinson's disease. One, the APOE on the right side here, was previously known to be a risk factor for Alzheimer's disease, and Susan will speak about that further. And then 2 were gene mutations or variations that had not been previously associated with one of the neurodegenerative dementias. So this still doesn't change our clinical approach to genetic testing, as so far these would be considered risk genes, but not deterministic, and Susan will speak to that as well. But it is just an example of how quickly advances are being made in these fields to help us understand what causes these diseases and hopefully to give us better ideas of how they can be treated. Now, the situation for frontotemporal dementia is a little bit different because a greater proportion of frontotemporal dementia does seem to have a strong hereditary component and a genetic hereditary component. So approximately 30% of patients with frontotemporal dementia likely have it because of genetic risk, whereas the other 70% are considered to be sporadic. And that means that there's only one person across many generations who's had FTD, and that the family history information is pretty complete so that we can be reasonably confident that truly there were no family members who developed the disease. For the 30% that are hereditary, there are 3 gene mutations that explain about 50% of the families where FTD is passed on in each generation. Those 3 genes are here, the MAPT in the red, granulin in blue, and C9orf72 in yellow. And I'll mention that the C9 mutation is also the most common cause of ALS, also known as Lou Gehrig's disease. Much less commonly in rare individuals and rarer families, there are over a half dozen other genes where mutations have also been identified that can cause frontotemporal dementia. Now, in those 70% of patients who are thought to have sporadic FTD, we know from research studies that most of them do not have any identifiable genetic mutation. However, about 6 to 8% do end up having a mutation that is found, most commonly in one of those big 3 gene mutations that we saw were associated with FTD. With that, when we think about the role of genetic testing for a patient with FTD in the clinic, we're focused on patients who have symptoms or a diagnosis of FTD. And because the heritability of FTD differs according to different FTD subtypes, we also think about the clinical subtype. So we do offer genetic testing to all symptomatic patients with behavioral variant FTD and with the non-fluent primary progressive aphasia version of FTD because of that 6% who may appear to have a sporadic inheritance pattern, but could be found to have a mutation. In contrast, we only offer genetic testing if a clear family history is present with other people with FTD, if the patient is presenting with the semantic variant of FTD, with progressive supranuclear palsy, or with cortical basal syndrome, because these 3 clinical subtypes are much less commonly running in families with FTD and are much less commonly found to have a known genetic mutation. So up until now, we focused largely on clinical genetics of symptomatic patients. In the next part of the talk, as a transition, Susan will also be talking about predictive risk and predictive genetic testing. In genetic forms of FTD, and the same is true for Alzheimer's disease, in the research realm, much of the focus is now looking at what are some of the first changes that we can detect in individuals who are in pre-symptomatic stages, so who may be carrying a gene mutation that will later on in life cause dementia, but who for the present time are functioning normally. Through the GenFi study, which we run here in London, as well as in other sites in Canada and in Europe, we're looking at this transition from being pre-symptomatic with an FTD gene mutation to the symptomatic stage. And both in our field in FTD as well as in other neurologic disorders, there's increasing interest even as to whether there may be some important lessons to learn in studying youth from families with genetic forms of these diseases. So we are launching this year the GenFi neurodevelopmental study that will hopefully demonstrate what is happening in the cells of children that allow them to compensate for the presence of that gene mutation, which is there, as you saw, from the time they're created. And might the processes that happen in their cells give us clues to help extend that so that they can remain asymptomatic, hopefully their whole lives, but certainly later on into adulthood than we can do right now. So with that little opening teaser on predictive and pre-symptomatic genetics, I'll turn it back to Susan for the third part of the remarks today.
All right. So I want to touch on discussing predictive genetic testing and some of the counseling issues at the very, I'll talk about the counseling issues at the very end. I think it's an important distinction to make between diagnostic testing, which is the sort of testing that Dr. Finger would do, or other neurologists when they have a series of symptoms and they suspect a diagnosis and they either want to confirm that, or they want more information, or there may be an advantage to knowing a specific mutation related to treatment. When we get referred a family to us to medical genetics, it's usually an asymptomatic, unaffected family member. And the most common referral that we get would be a family member has Alzheimer's disease, or a couple of family members have Alzheimer's disease or dementia, and they would like genetic testing. Unfortunately, there's nothing we can offer them. And so we decline those referrals, and people often wonder why. And there's this misconception that there's a genetic test for Alzheimer's disease. There is not. As Dr. Finger pointed out, the vast majority of Alzheimer's disease is late onset. Of that late onset, only about 15 to 20% of it is actually familial with 3 or more cases. And early onset familial dementia is probably less than 2% of families. And the reason that's important is it means that when we're talking about these genes that Dr. Finger introduced to us, it's important to understand that genetic testing, where we can offer predictive genetic testing, only applies to these 3 genes, because these are disease-causing genes. And so that's a very small proportion of the population. And even of the people who qualify for early onset familial Alzheimer's disease testing, still 20 to 40% of those people we're not going to find a genetic mutation in. So it's really a small number of families that we're even able to offer predictive genetic testing. So in referrals for genetic testing for Alzheimer's disease, we can only offer it if a known mutation has been identified in an affected family member. The same is also true for frontal temporal dementia, where these are the main genes. Again, majority of causes are unknown. They're not necessarily mutations in these genes. And so again, we can only offer genetic testing when a familial mutation has been identified in a familial family member. And the 2 main genes for Lewy body, majority of people who have this condition are not going to be identified with mutations. So again, there's very limited predictive genetic testing available. A really important concept to also understand when we're talking about predictive genetic testing is the concept of penetrance. And it's a term we use in genetics to mean if you inherit a specific genetic mutation in a disease-causing gene, what's the probability that you're actually going to get symptoms of that disease? And so the first gene that we were able to ever do predictive genetic testing for was the Huntington's gene, which was found back in 1993. People who have the Huntington's mutation, it's 1 gene, 1 mutation, and it's 100% penetrant. So it is appropriate to offer predictive genetic testing because you're able to say to that person, if you inherit this mutation and if you live long enough, you will develop Huntington's disease. So in order for us to offer predictive genetic testing for any particular gene, we also want the penetrance, or the likelihood that that person is going to be affected by that condition, to be 90% or greater. Another important distinction when you're talking about genetic testing is the difference between a disease-causing gene and a disease association gene, or what Dr. Finger would have referred to as a risk gene. So in a disease-causing gene, a single mutation in that gene can cause that person to have the disease. In a disease association gene, a mutation in that gene increases the likelihood or the risk factor for that person to develop the disease, but it doesn't necessarily mean they're going to develop the disease. And for Alzheimer's disease, over 20 genes have been identified as risk genes or disease association genes, but the majority of them don't in and of themselves necessarily increase the risk specifically a lot. The exception to that is APOE. And the problem we have in genetics with APOE is people think this is the Alzheimer's gene, that if you can have genetic testing to determine if you're going to develop Alzheimer's disease by testing for APOE. It is true that APOE is the strongest of the risk association genes for Alzheimer's, but it is not a disease-causing gene. And it provides the code for this gene provides the instructions for making a protein called apolipoprotein E. And there are 3 different versions of this gene that have been identified: APOE2, APOE3, and APOE4. And I kind of like to, so it means they have slightly different genetic codes, but essentially they're creating a functional protein at the end of it. And when we use the term protein in genetics, we're not talking meat, fish, eggs, we're talking a molecular structure that has a function in the body. So when I'm thinking of the variations in APOE3, it's kind of like, okay, I drive a Subaru Forester and it's gray. Somebody else might have one that's orange. Somebody else might have one that's blue. There are different variations of the same car, but they all function. And so that's kind of what the variants are like when we're talking about different gene variants, or what we refer to in genetics as alleles. And so APOE3 is by far the most common in the population. APOE4 comes next, and APOE2 is much rarer. Now remember that we have 2 copies of every gene. So you can have various combinations of these versions of the gene. And people have been identified when they have APOE4 as having an increased risk factor for developing Alzheimer's disease. So when we add, but that risk factor is lower than previously thought. So again, there's one of the problems with an association gene is the statistic that you can provide somebody with when you do genetic testing for something like APOE is only as good as the study you last read or the information that you've been provided with. So one of our genetic resources, if you have 1 copy of APOE4, so your other copies may be 2 or 3, then you have a 10% to 20% risk of developing Alzheimer's disease by age 75. If both of your genes are APOE4, then that risk increases to up to 35% by age 75. It's important to keep in mind this gene is also not useful for diagnosis because 42% of people with Alzheimer's disease do not have an APOE4 version of this gene. They have E2 or E3. And so it doesn't rule out, sometimes neurologists choose to do it in adjunct to other testing to help facilitate diagnosis, but often it's not useful diagnostically either. APOE2 appears to actually have a protective effect. Unfortunately, this protective effect is overtaken by the risk effect of APOE4. So if you have the genotype A2 and 4, you would think maybe they would cancel each other out, but unfortunately they don't. I took this from a study in the Canadian Medical Association Journal in 2018, and they have slightly different statistics than I have. For somebody who is E4, they're saying that the risk for developing Alzheimer's is 19 to 24% by age 80, and for any dementia, 33 to 38% for all dementia. And the purpose of this is to highlight the fact that because it's not diagnostic testing, you're not going to ever get an accurate assessment of what your risk is because it's based on a conglomeration of various studies, and that's only as good as the study was originally. So this testing can give you a statistical figure, but that statistical figure is going to have a range to it. The other important sentence in this is the present absolute 10-year risk estimated by dementia by age, APOE3 have potential to identify high-risk individuals in targeted prevention interventions. And I think where the carrot is being dangled to people is saying, this is one way that we can identify people who are at high risk. I would be hesitant to use the word high risk because you're qualifying the risk. Some people would consider a 20% risk okay. Some people would consider it high. Researchers what they're essentially trying to do is find a population the way Dr. Finger is with the FTD population, but they're using disease-causing mutations to identify that population. But to find in the late onset Alzheimer's community, a population that they can then study and potentially find interventions. So if you're considering genetic testing for APOE3 or for the APOE gene, you need to consider why you're doing that testing. If you're doing it to determine what your risk of developing Alzheimer's disease, it's not going to give you an accurate estimate of what that risk is. It can potentially produce a number, and any labs offering that testing will give you a number based on whatever their database has indicated, but it doesn't really tell you, are you going to develop Alzheimer's ultimately? And so when we go back to that penetrance, it's nowhere near that 90% that we would talk about in a disease-causing gene. So where are we going from here? How are we going to make that better? Well, in terms of the causes of dementia, for the majority of cases, they're polygenic or multifactorial. And when we say polygenic, it means it's controlled by 2 or more genes. And when we say multifactorial, we mean there are many factors, both genetic and non-genetic, be they environmental or other non-genetic factors. And so how can we potentially use this moving forward to try and better understand people's risk in these risk groups that it's clearly not caused by a single gene disease-causing mutation? And so we all carry thousands of genetic variations, and typically single DNA changes can potentially be used in what we now call a polygenic risk score to modify people's risk and improve our ability to predict risk. So if we go back to when we're talking about single letter replacements, what we're saying is it's like if we go back to our original sentence, the cat ate the rat, the cat ate the bat is a single letter genetic change, and these are called SNPs. And so what's starting to happen is people in the researchers with Alzheimer's and other diseases that are most likely caused by are developing things called polygenic risk scores. And so they're taking populations of affected people with a diagnosis of late onset Alzheimer's disease, looking across the whole genome and essentially looking for hotspots that they're trying to find. So let's say in your research group, everybody in your research group, you found the variant, the cat ate the bat. Well, that would be pretty lucky. Research never works that way, but it means now you have an association of a genetic change in the code of that gene that may increase the likelihood of that person having developing Alzheimer's. So the hope is that as these polygenic risk scores improve, and in combination with potentially APOE testing, because it has such a high association, there may be an improvement on our ability to predict who potentially is going to develop symptoms of Alzheimer's later in life. And this is just an example of what a SNP looks like, and it goes back to that kind of, it's a different variation in the genetic code. It doesn't in and of itself cause disease, but it potentially increases the likelihood that you could develop that disease. And probably the most common example where SNP technology is used is when people do ancestry testing. And so when you're doing ancestry testing, what the company is doing is they're taking a sample of your DNA, they're looking across your whole genome, and they're identifying different SNPs. And so this group of SNPs means if you have that proportion of, those particular numbers of SNPs, you're more likely to have come from England. If you have these SNPs, then you're more likely to have come from Greece. And so it's a good estimate, but it's only as good as the database they're using. And so, for example, with Ancestry, if you do Ancestry, if you log into your results, it will change over time, because as they change their database and add in more SNPs, it potentially changes your risk estimate. So if you think about that for doing disease testing using SNPs, you need to know that the database that was created to come up with those risk estimates is really robust. And one of the companies that uses SNP testing to do direct-to-consumer testing, the most common one people have heard of is 23andMe. And so what you have to keep in mind when you're using those sorts of things is if it says your risk of developing stroke is 4 times the population risk, that's based on their data in their database. If you used a different direct-to-consumer testing who has a different database, different SNPs, it might not give you the same results. So there's a lot of work to do, but there's also a lot of interest in polygenic risk scores because it's another potential way that we can test for genetic things. How am I doing for time? Okay, so for those small percentage of people that potentially are tested by Dr. Finger and a genetic mutation is identified in an affected family member, that then offers the option of predictive genetic testing for, or what we would call cascade screening for all other family members. I think it's important to keep in mind that just because you can then test unaffected family members doesn't necessarily mean that you should, or that people even want to have that information. So when we look at, again, at our Huntington's population, because that's the one that we have the most experience with, when we first started offering genetic test or predictive genetic testing back in 1993, only about 20% of people who are at risk choose to have testing. And that number has held true today, that the majority of people who know that they're at a 50% risk of inheriting this mutation choose not to have that information. And one of the things I found very interesting is a public broadcasting company came out with a movie about trying to decide called Do You Really Want to Know? And they had a little ticker on their website when they were advertising it saying, would you want to know? And so I voted to see what the response was, and 80% said they would want to know, and 20% said that they wouldn't want to know, which is the exact opposite of what reality is. And what's interesting about that is I think when you're not in the position of actually having to make that decision and know that you're at a 50% risk of inheriting a gene that causes a disease for which there's currently no treatment, you want to get tested because you want to know that you don't have it, but you haven't done the homework or the soul searching or the thinking about, but what if you're the 50% who do have it? What's the impact for you? How do you value that information if you decide to go ahead with the testing? What's the potential harm in terms of effect on self-esteem, effect on future outlook on life? Balanced again, what's the value? Will it help you make decisions about retirement, about financial planning? We also talk to people in these sort of counseling sessions about the timing. So we don't test children in predictive testing because a parent shouldn't have the right to make that decision for a child. An adult should make the autonomous right themselves to know if they want to know what's in their future. But the timing of the testing can vary. Just because you find out that there's a gene mutation in your family doesn't mean you need to rush out and get testing. What we know and understand about these diseases currently is going to change over time. Our ability to potentially treat these diseases and do gene therapy may change 20, 30, 40 years from now. So if you're an 18-year-old getting tested, by the time this disease is ever going to affect you, that information will have changed radically. And do you necessarily need to know that information when you're 18 versus when you're 30 versus when you're 40? People also don't necessarily think about the implications to other family members. So any positive genetic test you get, now all your first degree family members are at increased risk. And we also talk about, what else is going on in your life. These are life-changing events to do predictive testing and find out you've inherited a gene that's going to cause you to have an potentially untreatable disease in the future. So those are just sort of briefly some of the things that we talk about in genetic counseling, and we can certainly expand on it in the future. The key things to understand is for the majority of cases of dementia, genetic testing doesn't really assist with family screening and diagnosis. There's a lot of people think genetics is very black and white and will give you concrete answers. The majority of genetics is actually very gray. And accurate predictive genetic testing can only be offered when to unaffected family members when a familial mutation has been identified in an affected family member in a disease-causing gene, and there's high penetrance. I should probably have added that in as well. So I'll finish there and go to questions.
Thank you very much, Susan. Dina, turn it over to you, I think, to moderate. And if there are no questions in the Q&A or when we run out, I have a question for Susan as well.
Thank you so much, Dr. Finger and Susan, for such an informative and helpful presentation. To our attendees, I'm sure you have many thoughts and questions after hearing from our panel. We have approximately 13 minutes available for questions, so we will do our best to answer as many as we can. So just a friendly reminder to everybody, if you do have a question, please add them to the Q&A box, not the chat box, so we can keep track of things. And also just a friendly reminder to watch to see if there's any common questions that you have, so you can upvote your question as well, so it gets moved to the top of the list. So I'm just going to open up our Q&A box here. I'm just going to stop my share here for a minute. Okay. Oh, sorry. All right. So I'm just going to pop up the Q&A box here. For some reason, Susan, I'm not. Oh, here we go. Okay. So we have a comment here that says, excellent presentation, thank you. And it says, our next question is, for individuals and families who do qualify for genetic testing and are interested in exploring it, what can we tell them the process is like? Where do they go? What is the process like? How long does it take? Oh, Susan, you're muted.
I'm still muted. Yeah, sorry.
Yeah, sorry, that's all good.
I keep losing the mute button. It keeps moving around my screen.
No, it's all good.
So it's different in our genetic center. We had a predictive testing protocol for Huntington's, and we've adapted some of that for all predictive testing for adult neurological conditions. So the place that you would be referred if there's a known familial mutation in the family would be to a medical genetic center. And testing protocols do differ. Most of us require at least 2 counseling sessions. It's been a bit more difficult with COVID because we've been doing all this over the phone. And when you're talking about difficult decision making, it's a little bit easier to do in person. So sometimes we do it by video, and certainly we do results by video. And that's true for a lot of different centers. I can't really tell you how long it takes because different genetic centers have long wait times, but this is a London group. My wait time is only a couple of months. So if you're being referred to our medical genetics unit, the process is usually relatively straightforward. If the person in your family who is affected hasn't been tested, then they need to talk to their neurologist about whether or not they would qualify for genetic testing to see if they can identify a genetic mutation in the family that then could be used for genetic testing. Hopefully that answers the question.
Thank you so much, Susan. The next question is, can individuals obtain their own testing, and how much does it cost?
I don't know why I keep hitting the wrong mute button.
Oh, it's all
yes.
good. Okay.
Yes, there is. So it depends what you're testing for. In terms of clinical genetic testing, there are companies that through Life Labs that you can order the testing through your family doctor. The cost generally depends on the panel that you're ordering or what you're specifically interested in. So whether it's an Alzheimer's panel or an FTD panel or just a dementia panel, the size of the panel sometimes changes the cost. Life Labs uses a company, Centogene. I think their panels are in the $1,000 range. There are companies in the States where it may be cheaper, or there may be what they call free or sponsored testing. I caution you about free testing. Nothing in life is free. When you do sponsored or testing for which you don't have to pay, they own your DNA. They own your sample, they own your result. They can then share that information or sell it to pharma, which may or may not be a bad thing. If people aren't doing research, we'll never move forward with information, but it's an important thing for people to know if they're worried about privacy or what happens to their DNA sample. When we order clinical genetic testing, the sample is essentially destroyed unless we have specific reasons to want it banked.
Thank you, Susan. Great presentations. I'm wondering what factors influence penetrance. Could there be protective effects of other genes or environmental factors?
Yes, there's definitely modifier genes. So we have another term in genetics called variable expressivity, which basically means the same disease can look different in different family members, even with the same genetic mutation. And so we know that there are modifier genes that can potentially improve things for people or potentially make things for worse. Probably the best example is of variable expressivity is the C9orf72 gene, which causes both ALS and frontal dementia in the same family, or in some unlucky people, they get both. And so that's the same mutation in the same family, but causing a different expression. And we see that in a lot of dominantly inherited conditions, not just in neurology. And so we know that there are probably modifier genes. We just don't have any way to test for them currently. And polygenic risk scores may help with that as well.
Thank you, Susan. Are there any tests available to people who do want to predict their likelihood of developing dementia, and then in brackets, who do not qualify for genetic testing when a parent has had dementia?
Yes, there's direct-to-consumer testing. I caution you to know what they're actually testing for, and there are private companies that are also offering disease-causing testing and consultation. So there's MedCan in Toronto, is one where you can pay for genetic testing.
Thank you. In lieu of testing for Alzheimer's, what steps can a person take to reduce environmental factors?
Sure, I can take that one. A lot of the risk factors that seem to reduce the incidence of dementia really reduce the risk of the vascular component of dementia, little strokes and bleeds, what can lead to vascular cognitive impairment. Sometimes that may interact with Alzheimer's disease, so we generally give this advice to everyone, but it's to make sure that you exercise, ideally that you eat reasonably healthy, and the Mediterranean diet is the one in vogue now that is associated with perhaps later onset of dementia. However, the caution there is that is not causal evidence. Nobody has been randomized as a baby to one diet or the other, so we don't know whether that diet really helps, but it does seem perhaps to be seen in populations with lower incidence of dementia or later onset. Staying cognitively and socially active are other key aspects. They exercise networks of the brain, just like we exercise our muscles, so they can help build compensatory connections, sort of backup connections, that if some of your brain cells stop working in early stages of dementia, you have others that can pick up the slack and therefore delay the appearance of your symptoms. Making sure you're not smoking, trying to eat well and avoid diabetes, or if you have diabetes, having good blood sugar control, controlling blood pressure and cholesterol, all seem to be important for keeping the blood vessels in the brain healthy and reducing their contributions to cognitive impairment. Other risk factors specific to Alzheimer's disease are less well understood, but one maybe with a bit more evidence is to try and avoid getting concussions if you can. So wearing your helmets and being mindful of things like boxing, which really are associated with an increased risk.
Thank you, Dr. Finger. Should an adopted person do Ancestry to help potentially find their medical history, or whether they come from a long living line?
Won't help. Sorry. All Ancestry will tell you is where your genes came from. So what was the country of origin? It won't tell you if any of them are defective or don't function. There is something like 23andMe, which does SNP testing, but remember, I would caution that it's only as good as the database that they base it on, and it's sort of disease association testing, not disease causing. So all it will tell you is you have a 4 times more likely chance of having stroke. And I had a patient who phoned me in a panic, one of my cardiology patients, saying, I just did 23andMe. I know you'll be mad at me, but I know I wasn't. But the results show I have a 4 times higher stroke than the average person in the population. And I said, but Mary, your mother had a stroke, your sister had a stroke, you have 2 cousins who had a stroke. I could have saved you $99. You do have a 4 times more likely chance of having a stroke. And so the information that you're getting from that is relatively limited. It's risk association, it's going to be percentages. And the question is, will that affect your medical management? No, it won't affect how your family doctor treats you. Will it affect your lifestyle? Potentially, if you know that you're the sort of person that's then going to change your lifestyle. But we should all be doing what Dr. Finger just said we should be doing, regardless of our, our ancestry or, what faulty genes we may have.
Thank you, Susan. My son got 23andMe testing and they identified the APOE4 gene. His dad has recently been diagnosed with dementia, probably caused by Alzheimer's. His dad's father had Alzheimer's and his great-grandmother also had some form of dementia. Any advice for my son?
Susan, do you want to start on the genetic piece there, and I can add in from the clinical side too?
So again, it goes back to statistically, what's his chance of actually developing Alzheimer's? And we know it, it may be 20, 30%. And so the question is that valuable information for him to have, or has that just created anxiety without knowing whether or not you're potentially going to, this is the downfall of doing this type of testing, is you now have information that has potentially a negative impact on you, and there's not necessarily anything from a positive perspective that you can do. Just because the family history is positive does not increase the likelihood that your son will develop dementia, because there's already dementia in the family that there presumably is a component that the APOE4 contributed to.
And it would be the same, same recommendations as Susan mentioned, that we would say for everyone, to stay active physically, cognitively, and socially. And then in this case, to pay attention, but in communication with your family doctor, not now, but maybe 5, 10, 15 years from now, there will be something to modify if you've got the APOE4 gene. It's just not there yet, but we would hope down the road that might be developed and useful. But right now it isn't, so we need to stay tuned.
Thank you, ladies. Okay, we are just about at time here. We've got about 1 minute left. If anyone has any other last minute questions, please throw it in a Q&A right now before we start to wrap things up. Otherwise, the timing is worked out really well. So thanks again, ladies. I'll just wait here to see if there's any last minute questions. Okay, so let me just share my screen again here. So thank you again to RBC Wealth Management Dominion Securities for generously sponsoring this event. Dr. Finger and Susan, thank you so much for taking the time out of your busy day and your schedules to share your knowledge and expertise with us. I know personally for myself, you've helped clarify some things that I was thinking about, and the way that it was presented, it was something that I don't have a science background, so I was able to understand that. So thank you so much. We know your presentation will stimulate further conversations. To our attendees, thank you so much for attending this month's Brain Matters webinar. We hope to see you for our future webinars. And I'm just going to, here. On February 5th, 2022, we will be hosting the Cabin Fever Reliever event. It's back. This year, we are offering 2 ways to attend: in person at the Best Western Lamplighter Inn and Conference Center, and virtually from the comfort of your own home. For registrations, please go to our website, and we will continue to monitor the situation through public health guidelines for the in-person event as this may change. Lastly, please take a short evaluation on your way out when you leave the webinar today. It should pop up on your screen when you click the Leave button, or you can scan the QR code using the camera on your phone to access the evaluation. Thank you again for attending. We would like to take this opportunity to wish everyone a safe and healthy holiday season. We look forward to seeing you in 2022. Enjoy the rest of your day, everyone. Thank you for coming.